Sean, let me know if we're okay. And welcome everyone to Wine Wednesday with the Whisperer. We have some Pinot Noir tonight, which is my new favorite thanks to Colby. And I have a very special guest. So we won't spend much time on the wine. We're going to get right to the guest. I know that was a
I don't mind.
that was a quick through. Well, okay, actually, before I even introduce you, my first question is what is your favorite wine?
I drink a lot of Pinot Noirs and Malbecs.
Okay.
Um, yeah, especially from South Australia.
Is there a particular, brand that you prefer?
Honestly, by the time I have a few glasses, I don't care what the brand is.
You are welcome back on Wine Wednesday anytime. Okay. Anyhow, uh, for those of you who don't know, since I started ahead of myself, behind schedule as always, my guest today is none other than Graham Short. And welcome, Graham. I'm
Thank you very much. Thanks for having me.
absolutely. We're going to talk about some really cool stuff, so I won't keep you waiting. I'll jump right into it. Um, tell me a little bit about yourself, Graham. I know, but tell the audience what you do for a living and how you got there.
So, I am a research associate at the California Academy of Sciences, and I'm in the ichthyology department, and which makes me an ichthyologist. An ichthyologist is basically someone who studies the taxonomy and systematics of fish.
Okay.
And the fish I study are all the fish species in the Synathidae family, which means pipefish, pygmy pipe horses, sea dragons, and seahorses, and every oddball in between. And I've been at the academy for since 2005 now. Um, I'm involved in various projects, but we can get in that later. But
Yes,
that's just the intro.
yeah, I know my questions don't allow you much of an intro, do they?
Oh, it's all right.
Well, tell me this: what you basically explained what the ichthyologist does, but I'm curious, what made you decide to focus on Signathidae or Signathids? I'm sorry.
So it did start in about 2003. Um, I started living on my boat in Australia
Mm-hmm.
in 2003, 2004, and started doing a lot of scuba diving in the various harbors and bays. And when I saw my first weedy sea dragon, which
Oh
is a
wow.
cousin to the seahorse, and just absolutely fell in love with them. And then I decided, well, I want to start doing some research on these species. I didn't really know at the time who was doing what. So during the course of those investigations, I had met my colleague and friend, Dr. Healy Hamilton, who was at the California Academy at the time. And she studied Hippocampus cuda throughout the Indo-Pacific. And we talked about some collaborations together, and we decided to study the entire family. Because at the time, no one knew which species was related to which.
Right.
So we decided, well, let's collect at least all the genera that we can, and we'll collect most of the species later. So 10 years later, we found about 95% of the genera within the family. And basically, behind the scenes, that entails huge field collecting trips in Australia, all around the South Pacific, the US, Europe, and elsewhere. Um, so it's really thanks to the weedy sea dragons I got into the Cygnathidae family.
I'm sorry, I did not at all mean to leave you hanging. I was listening. I was having a teensy bit of an audio problem. Do I just talk louder or what?
I can hear you. I can hear you fine.
See, he can. Well, I had somebody checking the. You know what? I'm taping it so it does. If people can't hear me, then you'll hear it in the replay. Okay. um,
That's what you have editing for.
that's right. Live streaming is not my forte, but we're still gonna continue. So, can you tell me? what is the process for determining a new species? I know you were talking about the weedy sea dragons. I'm sorry to jump around, but
No, not at all.
what is the process? What do you go through to actually qualify a new species when you're out there looking at them?
So there's a lot of there's many levels you have to reach in order to probably describe a new species. Um, I mean, for example, it can come from many angles. If I were to look through a museum collection and if I see something unusual, I'll take that specimen out, put it under a microscope, and I'll do trunk and tail counts. Let's say if it's a seahorse, I will look at the spines. So the presence or absence of spines and the position of the spines in a seahorse is very much like a human fingerprint. Each one is unique to a seahorse species. And so let's say if I see, wow, the spines are different, seahorse looks a little bit different, I need to do some further research on it to corroborate that's a new species, I would try to find one in the wild.
Okay.
So that entails doing a field collection trip, obtaining a specimen, and then bringing it back to the lab to do a genetic analysis on it, just to really clinch the deal. And so basically, we sequence the mitochondrial CO1 gene, or just a partial fragment, and that's like a barcode for animals.
Okay.
And so you compare barcodes. Let's say I get the barcode sequence and I compare against other seahorse barcode sequences. And right away, I can tell whether it's different or not. And so if it is, then I can say I definitely have a new species. And then the process of describing it is you have to write out in excruciating detail every single thing that makes that seahorse new. So you write a diagnosis and a description. The diagnosis is what separates that fish from the other seahorses. Then the description entails writing every single wart, nook, and cranny of that fish. You know, the spines, replacement of the spines, meristic, morphometrics, genetics. And then you submit it to an academic journal. So there's really like 3 or 4 big steps,
Mm-hmm.
and it takes a little while, but it formally establishes a new species.
Right. I mean,
Yeah.
I think it's awesome. I am planning to ask you about a few other things that I did see that you wrote. But when you said the genetic part of it, is that the DNA? What's the word I'm looking for? The DNA
Yes.
sampling. How does that work?
Yeah. So basically, you can take a little fin clip or a bit of the spine or a little bit on the right side of the body of the fish, and then you extract DNA using the DNA extraction kit,
Mm-hmm.
and that just takes a few hours. And then once you have a clear solution of genomic DNA, then you do what's called PCR, or polymerase chain reaction, and you amplify a second of the mitochondrial CO1 gene. And that means you produce millions of copies of that gene, which you can run through a gel and you can visualize it as a band.
Mm.
You cut that band out, you bring it to someone who can sequence it, and then you compare those sequences against other seahorse sequences. And you can so once you have the sequence, then you can play around with it on your laptop using various software.
Mm-hmm.
So once I had that sequence from that putative new species of seahorse, you align it with other seahorse sequences, then you can see the differences or not.
That's really cool. Go ahead. Go
Yeah.
ahead.
Oh yeah. So and then you produce what's called a phylogenetic tree, and you can see where that seahorse falls with other seahorses. So it tells you right away, well, this seahorse might be more closely related to Erectus or to Hippocampus patagonicus. And then you just go from there. That just adds to the body of data in your paper.
Gotcha.
Gotcha. Yeah.
Oh, now I'm not gonna.
You know, I made a big mistake. I thought this was Tequila Tuesday.
Oh no, it's okay. You know what? I'm gonna tell you a little secret. A little secret is that I typically mess up a lot, and as I'm, doing my Wine Wednesday, you'll see me grab a Ricker's cup with which might or might have vodka in it. But we're good. We're good. Wine Wednesday is just the chill
Cheers.
place for us to talk and for you to teach us and so that people can hear about these fascinating things that you're doing. And when you were talking about the differences, we're going to jump around a little bit. I tend to
Yeah.
do that. But when you were talking about, I followed you with the lining it up and comparing with other, already lined up genes and all that good
Yeah.
stuff. Um, is that and then you also spoke about how you could then determine where they came from. I know you've done a lot of work with that, and was that I guess what I'm asking is tell me kind of about the story you told me yesterday, and was that about the pipe horse? Tell me the difference and all that.
Okay, so right now, so I just published a paper on Hippocampus jabipigu. It firmly established that the Japanese pygmy seahorse is a new species compared to some other similar species of pygmy seahorse. But I'm also working on a pygmy pipe horse. And a pygmy pipe horse looked a lot like a seahorse, except the angle of the head is not as severe as that you find in a seahorse. In
Okay.
the seahorse, the head's usually angled up to 90 degrees. In a pygmy pipe horse, it's usually around 25 degrees. Um, so they look very similar. They both have a prehensile tail, brood pouch, and so forth. Um, so this pygmy pipe horse was collected in New Zealand
Mm-hmm.
by me and some colleagues. And the last signatha to be described in New Zealand was in 1911. So more than 100 years later, here we are with a new signatha species being discovered in New Zealand, which is temperate. You have the North Island and the South Island. So it's very much like going from Central California to Seattle in
Right.
terms of water temperature. So it's very exciting. Excuse me. So during my course of investigations of this pygmy pipe horse, I found out that not only was it a new species, but it's a new genus. And I found this out by looking at certain diagnostic morphological characters present on the fish. And it looks very similar to species of temperate pygmy pipe horses from southern Australia.
Okay.
In fact, it's almost identical superficially. You look at it like, oh, it's nothing different. But when you look at it closely, you'll see some differences. Primarily, and this is the fun part, this pygmy pipe horse has a coronet just like a seahorse.
Wow.
So it was found, it was seen before many years ago at the Poor Knights Islands, which is a remote island off the northeast coast of New Zealand.
Mm-hmm.
And they thought it was Hippocampus jugamus, which is a really rare seahorse found in the Southwest Pacific. So they looked at him like, well, that's just a seahorse. It has a coronet. But when you really scrutinize it,
It's
it's not a seahorse. It's a pygmy pipe horse.
That is so cool. And
Yeah.
is this the work that you presented at the Sing Bio
Yeah,
in
I presented
Tampa?
Symbio. It was a 5 minute talk, and I went over by 10 minutes. So
Yeah.
they're all wavy behind the scenes, stop, stop.
Right, right.
Once you get me going on Signathas, I can't stop.
Oh, but I mean, it's fascinating. Talking to you gives me such joy and hope because there's just so much we don't know. And we were talking yesterday, and it fascinates me how much we know now versus 20 years ago, and how much you're finding that I had no idea about. It's great. Um,
Yeah.
so now we've got the end. Okay, let's see. So we've got the sig, the pipe horse. I'm just gonna call it a pipe horse. Why don't you go ahead and tell me? Um, Actually, before I get into that, what did you think of Sing Bio? Was it pretty? What did you think?
It was awesome.
Mm-hmm. That's what I've heard.
Honestly, I didn't know what to expect because it had been a while since I've been to a conference. Well, this is the first time I actually met other people who were doing with similar interests, doing similar research.
Mm-hmm.
I met researchers who were studying breeding behavior, looking at ecological aspects of seahorses and seagrass. It was just a joy to meet people who are just geeking out on fish like I
Yeah.
been doing.
No, exactly. I meant I had tickets to go this past year or this past one, and I missed it. And so when I saw that you were at the one in Tampa, I was like, oh man, that's
I
so cool.
know it was fun because we had a great time. It was friends from all over the world. We finally met in person, and we went out a lot, and just geeked out on fish talks, and all the talks were amazing.
Right.
But the best part was making new friends and connections, and it was actually thanks to Dr. Rich Smith from the UK who works on pygmy seahorses, who led me to Hippocampus jugupigu because he had observed these in the wild himself at Hachicho-jima Island, south of Tokyo.
Mm-hmm.
And so from there, it led to a collaboration to confirm that that species of pygmy seahorse was indeed a new species. So thanks to SynBio, it led to a wonderful paper.
Right. And you led me into my next question because I actually shared an article about your finding this new species. And I had no idea that it was you until I found the little G short, and I was like, oh, wow, cool. So tell me, you just told me how that came about. Obviously, it came from your connections at SingBio. But what all did it entail? What was different about them in this particular case that let you say this is a different
Sure.
species?
So with respect to the background, Japanese divers have seen or observed this pygmy seahorse for decades, at least for 20 years. I've been to all these obscure Japanese dive sites and I saw pictures of Hippocampus jugupigu, or as the articles in the US are saying, Japan pig.
Yeah, well, when I asked the pronunciation, I was like, okay, so Japanese pigu, got it. All right,
Yeah.
go ahead, go on.
So it's been known to divers, but for some reason it's never been formally described by academia in Japan. It's been collected by ichthyologists, but they just never got around to describing it. So I said to Richard, let's just do it ourselves because I have the equipment to create scans of their skeletons to look at their uh, detailed interior morphology in depth and compare to other pygmy seahorses I have in my collection. So we managed to find a local diver in Japan to get us some specimens, the ones you see in the paper. And then I did a DNA extraction on a little piece of tissue. I did CT scans of their skeletons and of other pygmy seahorses as well. And what I found blew my mind.
Mm-hmm.
I was looking at certain bony structures on right behind their head, let's say on their neck or on their upper back,
Mm-hmm.
you were. And I found these weird wing-like spines that were kind of stretched out laterally.
Okay.
And so a certain group of pygmy seahorses have these spines, but either they have one set or two set. And this one had a double set where the others mostly had one. But it also had this weird ridge-like structure on its back. You can see in the pictures like a fine red line
Mm-hmm.
on its back, and no other seahorse that's currently recognized has that dorsal ridge. So it's going to be important for another future paper. I won't get into it now, but it's really a key morphological feature that distinguishes it from all other seahorses.
That's awesome. And
Yeah.
when you're talking, please understand that I'm not, I'm far from your level. So
Oh
just
no, no
help
worries.
me out really quick. I saw the little red thing sticking out of the head and the back. Is that what you're talking about when you say
Exactly.
okay. And is that
Yeah.
the important key thing that's going to help you determine?
Yeah.
Okay.
So, in to say it another way, all if you when I looked at many skeletons of seahorses,
Mm-hmm.
and on their backs, it's usually very flat, just flat skeleton on the back.
Okay.
With Hippocampus jugupigu, you have these weird triangular bony formations that forms the structural basis of that dorsal ridge. So you have three or four triangles in a row on the back, and then externally, when you see it live, it's that red dorsal ridge that you see.
That is so cool. And
Yeah, in fact, no other fish has that. It's
yeah.
really bizarre.
Now this was not on the questions, so you can tell me you can't answer it. It's fine. But I'm just curious, sitting here talking to you. Do you think that that's a sign that they might be an evolved species or a more prehistoric species? I'm probably saying it wrong, but do you think that's a sign of, or just that we hadn't found it yet?
We just haven't found it yet. It's
Okay.
called a derived character, meaning it's in the group of pygmy seahorses, but it's the only species so far that has that morphological character that is unique among all pygmy seahorses. So it means it's it sets itself apart,
Hmm.
and we I don't know why it has that. Some say it's sexual selection, but it could mean anything.
Right.
Yeah.
And you'll find
Uh,
out and tell us. I have a feeling.
oh yes. Oh yes.
And okay, let's see. I was trying to think. Oh, I just find this stuff so absolutely
I
fascinating.
know there's so much to talk
And
about.
I know you do have to. We don't have much time, so I'm going to speed it
Yeah, I
up
still have
here.
a long time.
Um,
No worries.
let's see. Da-da-da-da. Okay, just out of curiosity. Sorry, I had to check my notes. Um,
Sure.
just out of curiosity, this is just my own personal question to you because I'm just curious because you are the basically the scientific side of things, and I am more the hobbyist captive breeding side of things. And so the article that I read about the Japapigu, did I say it correctly?
You said it perfectly.
Woo! Okay, so the article I read actually brought up the fact that we would not see them in, the hobbyist spectrum. Maybe someday in aquariums or in scientific settings, but not ever in a hobbyist setting because of the size restriction on imports and exports. And I just wondered what you feel about that. How, whatever you want to answer.
With with respect to imports, I think the size restriction's irrelevant. People have tried at museums and aquariums to keep alive pygmy seahorses and breed them. They've gotten up to a certain point,
Mm-hmm.
but then it failed. It's very hard to feed pygmy seahorses because they're so small. You have to feed them baby zooplankton, and it works up to a point, but then they die off because they're starving.
Mm.
We tried at the California Academy of Sciences, and we there was two expeditions to the Philippines, and they acquired a pair of Hippocampus bargebanti, and I saw them myself. They were beautiful and they were tiny, but after 6 months, the poor things gave up. Um, so they ended up in my collection.
Mm-hmm.
So
Oh man. Well,
I know I shouldn't have said that to
no, it's
tequila.
okay. It's okay. No, because it's the truth. I mean, we have to learn somehow. I mean, you're doing great things. There's nothing wrong with what you just said. I'm with you. I'm like,
Yeah,
darn it, but I know that Richard Ross had a program where he was breeding some different pygmies. Not that I don't think that specific species, but or that, yeah, it would be species. But
yeah,
it's just I agree with you actually that the size limit isn't really it. It's the type of seahorse and what their requirements. Is
right. Exactly, because you know how hard it is to even keep a captive bred
right.
seahorse alive. You know, it takes a lot of effort, and it's crucial that you know what you're doing. With pygmy seahorses, it's a whole nother ball game.
Yeah, that's
Yeah,
when I saw the article, I thought, oh, that'd be amazing, but I'm not gonna be the one because I had no way. I would be too afraid. So, okay,
yeah, and plus seahorses are CITES protected,
right.
and pygmy seahorses, especially, are protected, and plus they're so hard to find that they wouldn't really make a good import unless you can figure out how to breed them, and that's probably going to be a long, long time.
Sure. And I
Yeah,
do believe the article was based on, scientists breeding them, not
yeah,
hobbyists.
exactly. Yeah,
I know the timeline is a lot longer when you're talking about, seeing them in homes or even in breeders, facilities, but I just always wonder because I mean, really, to be serious, a while back they were like you'll never see seahorses, in the hobby, period.
Exactly.
So,
Yeah. I mean, like all the larger seahorse species, when you see pygmy seahorses in the wild, all the males are pregnant.
wow.
Yeah. And I think the male we had in our special tank for the pygmy seahorses, it was pregnant as well. I think it's just a matter of keeping them alive by feeding.
Right.
Yeah.
Most things do come down to feeding with seahorses. I mean, I've done a lot of breeding interviews with breeders also, and it's always the
Yeah.
key point is keeping them fed the right proper nutrition. So we need y'all to figure it out. But okay,
Exactly.
so how many different since you started working with cygnethids, how many different species have you found? New species have you been able to prove that they were new species? Do you have any idea off the top of your head?
So right now there are about 41 currently recognized seahorse species.
Okay.
But it turns out of those species, there are a few that need to be revised taxonomically. So they're not seahorses.
Okay.
Yeah. But I've described one, which is Hippocampus jadpigu. There's Hippocampus hema from South Korea. Um, right now I'm working on a project. I'll be describing 3 to 4 new species of hippocampus.
Cool.
Yeah, from Southeast Asia. Um, and I'm just going to be I'm describing pygmy pipe horse, a new species of pipefish from Sydney Harbor, Australia, one from New Caledonia, and there's like 5 others. It just,
Wow.
yeah, I'm really happy that I'm able to work on these new species because I'm just learning so much.
Of course. And
Yeah.
really quickly, I do want to hear a little bit more about that. But um, when you mentioned that there are a couple species that aren't really correct,
Yeah.
why? How did that happen? Do you have any idea?
Well, it's because at the time they didn't have a suite of morphological characters to broadly um, detail, like to broadly say that's a seahorse or that's a pygmy pipe
Got
horse.
it.
Pygmy pipe horses and seahorses are so similar, but there's certain defining features that really separate the two groups. And at the time, they didn't really have the full suite of characters to define either one. So if they found a pygmy pipe horse, they would say, well, I think that's a seahorse because I don't know much about pygmy pipe horses. I'll just put it in the mold of a seahorse.
Gotcha.
Yeah.
Okay.
And it's a totally easy and understandable mistake. Not mistake, but just, it's easily confused.
Right. I mean, we correct things and get better with more research and new things. I mean, it's how
Exactly. We're just
science
adding
works.
more data. Exactly. Yeah.
And so, just out of curiosity, because I know personally that Lucy found a seahorse literally in Florida that could or could not be a new species. So, my question is if someone finds a seahorse that no one, that even scientific people are saying, wait a minute, that doesn't look like any of the others, do they contact you? Are you the guy? Or how do they go about, helping with that?
Yes. Well, I'm one of the guys. I definitely appreciated that Lucy contacted me. It's just a matter of Googling who is an expert on seahorse morphology.
Okay.
There are a lot of ichthyologists in the US and around the world. Um, but, ichthyologists study a number of groups of fish. I mean, there's certainly other ichthyologists besides me who study seahorses. There's some in Australia, some in the US, British Columbia, Canada. But I was really happy that Lucy contacted me because I'd just been working on seahorses, and it would be amazing if it's a new species.
It's okay. It's Wine Wednesday. You can say, I'm the best. So, yes, I'm the man. You contact
You
me. No,
know,
I'm just kidding.
I'm
Uh,
such the expert.
I,
It's obvious.
I love it. I love it. And now, if someone like, say me, I know you've helped me on the side anyways. I really, really, really love the document that you sent. Thank you so much.
Mm,
I'm
you're
gonna I'm
welcome.
gonna try to do you proud with it. But what I was gonna
Okay.
ask if someone wanted to know like the differences in how to tell a seahorse from how to tell which species I'm stumbling my words. How to tell which species a seahorse is, a part of, or if they in fact are a pipe horse or whatnot. Is there any good place online to find that information, or?
Unfortunately, the information online is scattered. It's not centralized,
Mhm.
but there are certain websites that are indeed useful.
Okay.
Fishesofaustralia.com.au is a huge resource. Um, DavidHarasti.com of Australia. But it really depends where the seahorse came from.
Right.
So let's say, like, for example, at the museum, we receive a lot of illegally collected seahorses or those people who try to sneak them in,
Okay.
you know, for pharmaceutical purposes. So we receive bags upon bags of dried seahorses. And so it's up to us to ID them. And the quickest way is just to do genetic analyses on them. Otherwise, I would be in my hundreds looking at all these fish under a microscope. And it would tell us right away which species it is, and then it will give us an idea where they came from, and then go from there. But a good place is the distribution of seahorses. Let's say it came from Mexico. Well, which side of Mexico? The Caribbean side or the Pacific side? And they're not that many species on, North America.
Hmm.
Um, there's what, four? So and then you have to acquire a specimen. That's the only way to truly ID it.
Sure.
Seahorse, yeah.
Okay. Well, I was just. How come I'm not going? Okay. Whoops. Anyways, it stopped. Okay. Well, let's. I will just for anyone just checking in. Thank you, Luz.
Kevin, someone is telling that your sound is quite low.
I know that's what Sean said. I'm not sure what the problem
Is it
is.
my sound?
It's me. It's
Oh, it's
me. That's why
you. Okay.
I meant to do a sound test earlier. I will edit this and repost it, guys. I'm very sorry. I family stuff. I just didn't have time to check, but I will edit it and repost it so that you'll get all of the information. I'll do one more thing to try to.
Can you edit out me drinking? Oh, I saw that. Seahorse Whisper adjusted your audio level. Oh my gosh. I mean, there's too many. Um, Really, it's going to be the new species of pygmy pipe horse from New Zealand. It is absolutely beautiful. Um, it has a very reddish white color pattern to it. It's found. So I found it with a group of colleagues from Auckland Museum on a vertical wall, completely exposed to the open ocean. It was very, um, lots of current. We were swept back and forth. We had to hold on to kelp, and then we had to scan the wall to look for these fish, and they almost look like nudibranchs. Yeah, it was just trippy. And underwater photographs really bring out the colors and its morphology. It's absolutely gorgeous. I just fell in love with it. But I have to say, with seahorses, I do love Hippocampus zosterae. Yeah, because I do. And that's a dwarf seahorse, not a pygmy seahorse. Yeah, yeah. Yep, got it. Yeah, correct. You bring up a good point, is you assume there's going to be in a seagrass bed in calm water in some estuary. That's not the case at all. You'll find seahorses um, living happily among a bunch of rocks to vertical walls, completely exposed to the open ocean, clinging to buoys. They're found everywhere. Yeah, that's a cool thing about them. Totally. Yeah. I mean, the exposed open diving? All right. Yes. Yeah, one is my friend and colleague, Dr. Healy Hamilton. She really introduced me to the world of seahorses. And she really pushed me in directions I never thought I'd get into. It's just genetics, morphology. So it was really thanks to her that my career in seahorses began. Then there's also my good friend and colleague, David Harasti in Sydney, Australia. He's an expert in the species Hippocampus wadii, which is very geographically restricted to New South Wales and now to southern Queensland. But he introduced me to the world of seahorses too in Australia, and it's thanks to him that he expanded my knowledge, to a degree that I didn't realize was possible. So really, two key people. Yeah, yeah. Do I what? Oh no, no, I don't. I used to, and I used to have a lot of cichlid tanks. So I love Lake Malawi and Lake Tanganyika cichlids. However, I live half the year on my boat in New Zealand now, so I live on a giant fish tank. Yes. One, the boat is my floating home, and two, I'm a summer chaser. So I'm in New Zealand from November to April, which is New Zealand summer. And there I do work with Auckland Museum and with Australian Museum in Sydney and South Australian Museum, but it's also my floating home. And when I'm back, I'm in the San Juan Islands north of Seattle. And that's where I work with the Friday Harbor Marine Labs. Yeah, I think that information is half correct. Certainly, seagrass habitats have been deteriorating due to development, climate change, and so forth. But then again, like for example, in southern Queensland, Australia, the seagrass beds have been expanding and contracting depending on the weather and other variables. So for 5 years, you'll have sparse seagrass beds, but then for the next 5 years, you'll have vast, healthy seagrass beds. They do recolonize. But then you have other cygnathids, pygmy pipe horses, pygmy seahorses, large seahorse species that you find that are not in seagrass beds, among the rocks, vertical walls, and so forth, places you don't expect. Um, there are certain villages in the Philippines that have been using dynamite to kill seahorses and other fish for sale, fishermen, bycatch, and so forth. And that's awful. That's a horrible practice. However, those same people are being re-educated as to the value of not dynamiting coral reefs, scraping seagrass beds, and so forth. It's better to have scuba diving tourists to enjoy the seahorses. So it's two steps forward, one step back. So there is definitely hope for sure. Yeah. Oh, thank you. Glad to hear. Yeah, you really hit on a good point. There's a lot of taxonomic confusion in the literature. It's all subjective. One researcher will say, well, this is this seahorse. Another researcher will say it's a different seahorse based on their knowledge and experience. So that's why Lori now did that global revision of Hippocampus, the article I sent you, in order to set the record straight of what's really going on. And because the reason there is taxonomic confusion is because seahorses are so highly morphologically conserved. They look very similar. Hippocampus erectus looks very similar to Hippocampus wadii in Australia. I mean, they're clearly different species, but they look very similar. So, yeah. So they did a good job of mopping things up, but there's still tons of work to finish. And so I've decided to look at each species, eventually each species, and clarify their taxonomy, meaning what really is that species you're finding on the west coast of Australia or east coast of the US.
That's awesome.
Yeah. I buy organic, but it's automatically organic because since I'm in San Juan Islands, it's a very rural county, lots of boutique or family farms. So all the salad I buy is straight from the ground to my mouth. Yeah, so it's delicious, it's fresh, nutritious, and I'm not going to grow a third eye from eating commercially grown lettuce. Yes. I know that's why there's box wine. Yeah. No, yeah. Yeah, Kelly, I think the next one's going to be what year are we? 2018? It's either gonna be next year or 2020, which is not too far off. Yeah, and I can tell already that we'll have a lot of fun. It, yeah. By all means. Sure. There she is brushing her teeth. Great. Yeah, I something huge, uh, but that will be for next year because I have 3 papers to write. So one is the pygmy pipe horse from New Zealand. One is the new species of pipefish from Australia. And then another is clarifying the taxonomic status of Hippocampus wadii in Eastern Australia. There's a very similar species in southern Queensland that was thought to be different based on the taller coronet, but genetically it turns out they're identical. So now I'm synonymizing or merging those 2 species together into Hippocampus wadii. Yeah. So a lot of papers this year, but then next year I have really 2 special papers I can't wait to talk about once they're published. It's gonna, I, I think Seahorse people are gonna hate me and love me. I don't know which. Yeah, awesome. Me too. Well, I will let you guys know once I get the genetic results back from, uh, from what she's found. And honestly, I wouldn't be surprised if there were new species we found on the east coast of Florida, because as I said, who is looking and scouring Florida for new species? And especially Lucy, who's been, I think she finds samples for researchers. I think that's what she does. And is this for Florida Atlantic University? Who's Nancy? Okay. And do you know which institution she belongs to? Okay. No worries. So but Lucy seems to be, she knows her seahorses, know where they're found. Um, but it looks like she knows enough to distinguish Hippocampus erectus from radii. And if she thinks this is a new species, it might be a good chance it is. Huge. Yeah, it is. Oh, I will. Yeah, no, I will. No, it's people like you that disseminates this wonderful information, keeps the excitement going in the Snathid world, and I'm really happy to have been on here. Thank you. Thank you.